is not seen often in the United States, it's still a problem worldwide, and scientists all over the world are collaborating and researching the disease in order to create a vaccine that would hopefully protect everyone. There are 3 current approaches being used to tackle the parasite. The first one is transmission-blocking vaccines, and these would stop the parasites in the gut of the mosquito. The second are the blood stage vaccines. These would tackle the parasite once it's left the liver stage and gone into the bloodstream. And the third are the liver stage vaccines. They would kill the parasite when it's in the liver of the human. The first type of vaccines are the transmission-blocking vaccines, and these would neither stop the person from getting malaria, nor would it lessen the symptoms. What it would do would be to limit the spread of future malaria by stopping the parasite from developing within the mosquito. So, what you would do with these vaccines would use antigens and insert them into the human body, and then the human would create antibodies, which then the mosquito would take up when it took a blood drink from the human, and the antibodies would then prevent the parasite from maturing in the gut of the mosquito, and it wouldn't then spread to the human the next time a mosquito bit a human. The second type of malaria vaccines are the blood stage vaccines. And in these vaccines, you would create an immune response using antigens that would cause the body to create antibodies that would target several proteins expressed by the parasite itself. So this vaccine would stop the parasite from binding to the blood vessel surfaces, and it would also cause the human to recognize the invader in the future. Stage vaccines are the last type of vaccines, and these are the preferred type of vaccines because you'd be able to stop the parasite at its earliest stage when there are least number of parasites in the body. So there are 2 different types of liver stage vaccines that are currently being researched right now. The first is the type that would create an antibody response against the surface sporozoite protein of the parasite. And an example of this is the RTS-AS01 vaccine, also called Mosquirix, that's being developed by GlaxoSmithKline Biologicals. The second approach is to create memory cells within the body using live parasites that are weakened. And this one example of this is the GAP vaccine, which is also the genetically attenuated parasite vaccine. RTS-AS01, also known as Mosquirix, is developed by GlaxoSmithKline Biologicals, and it's the leading malaria vaccine candidate right now. It's in phase 3 of clinical trials, and it's been tested in 7 countries in Africa. What it does is it's a recombinant vaccine. So it combines the circumsporozoite protein of the parasite with the hepatitis B surface antigen. It's inserted into the body, and it creates a cell-mediated immune response. And it gets the CD8 T cells to come to the site of the parasite and attack them and lyse them open and stop the reproduction of the parasite. The second liver stage vaccine that's being developed today is the GAP vaccine, the genetically attenuated parasite vaccine. And it's actually being developed right now at the Seattle Biomedical Research Institute. And this approach would use the weakened form of the parasites at the sporozoite stage, and you can weaken the parasites either by using radiation or gene deletion. Radiation is a little bit more difficult because you have to make sure that you radiate the perfect amount. If you radiate too much, you'll just kill the parasite and it won't be able to even migrate into the liver. But if you don't use enough, then the parasite will continue on its way to infect the human. But this process of weakening the parasite would create memory cells in the human that would cause the human to recognize the strong parasite when it enters the body later on.
A major challenge for limiting malaria is resistance. A long time ago we found out that if we kill all the mosquitoes in an area, it would become eradicated. So then we found the pesticide DDT. And DDT worked really well for us, so we sprayed it everywhere. But then the mosquitoes started to come back. So we went in for our second round of DDT, and we were expecting the same results. However, we then found DDT becomes ineffective very quickly. In fact, it only takes months for the mosquitoes to become resistant to it. We found our first specimen in India in 1959. Then we found the anti-malarial drug chloroquine, and it was created in Germany in the 1930s. But we first started to use it after World War II in the late 1940s because there was lots of cases of malaria breakouts. The first case of Plasmodium falciparum becoming resistant were in the 1950s. We have no idea where though. And since then, resistance has grown rapidly with the many different mosquito variations. Today, we found that drugs containing artemisinin-based compounds work really well. But in order to prevent resistance, the WHO has requested that we combine them with other drugs. There's many basic preventatives you can take if you're traveling to countries that have a reputation for malaria. Simply don't go to notorious areas. And mosquitoes like to hang around water, so maybe don't go to rivers. Especially don't go outside during nightfall. Make sure you wear preventative clothing. Use insect repellent, and bed nets when you sleep are the most basic ways.